Three, hold on. 3, 2, 1, and started.
Hello, everybody. My name is Jim Lamondia. I'm the chief scientist at the Valley Laboratory, and I would like to introduce our speaker today. It's Elisha Allan-Perkins. She was a postdoc with me in 2018, 2019, working on boxwood and on hop diseases. And she's going to share some of her information about a new pathogen that we discovered here in the state. The title is Discovery of a New Diaporthe Pathogen Affecting Hop in Connecticut.
All right, thank you, Jim, and thank you to everyone for attending this talk. So this is a project, as Jim mentioned, that I did during my postdoc at the Connecticut Agricultural Station. And the co-authors on this project are Dewey Lee, Neil Schultes, Samira Yves, and Jim Lamondia. So next slide, please. Can we advance the slide?
Yes, I did.
Okay. Okay, so Humulus lupulus is the scientific name for common hop. It's a perennial climbing vine. It's herbaceous. It's grown for the unfertilized female cone, or the hop cone. And this is harvested because it contains many important compounds such as resins, essential oils, and polyphenols. You can actually see the ligulin gland on it. Next slide, please.
Okay.
So those different compounds, those polyphenols and acids, those are all used to flavor beer. That's their main purpose for being grown. And they contribute to things like the bitterness of IPAs or the grapefruit piney flavor you might taste in certain brews. In addition to being really important for all these different flavors, and the different varieties contribute to different amounts of these oils and those different compounds, they also have antimicrobial properties that help preserve the beer. Next slide.
Okay.
And those antimicrobial properties have also been used in commercial products such as deodorant. And there also is some evidence that some of these molecules can be used for medicine, so for combating cancer or for HIV treatments. But still 90% of all hops produced are used for the brewing industry for beer. Next slide, please.
Okay.
So the hops are mostly grown between 35 to 55 degrees latitude. And for the longest time, Germany was the world's top hop producer, but the US has recently surpassed them. Next slide, please. And you can see circled on the slide, the Pacific Northwest is the major hop growing area currently in the United States. Next slide, please. So although they're currently grown in the Pacific Northwest, historically there was hop production in Connecticut. So hops were brought to the United States, to Massachusetts in 1629, and that was the Humulus lupulus variety lupulus, which is the hop we use in the brewing industry. There were some native hops already here, so that's Humulus lupulus variety lupuloides and variety pubescens. But most of the hops that are used in brewing are the European variety that was brought over. And these hops spread throughout New England and New York. However, with an increase in downy mildew pressure because of our nice, cool, wet springs, and also the start of Prohibition in the early 1900s, hop production really decreased in the Northeast. And that's when it became more dominant in the Pacific Northwest. But because we do have this history of hops, we know hops can grow here and they do well here aside from downy mildew. So there's interest in growing them here again. So next slide, please.
Sorry, just to interrupt. Elisha, which slide are you at? Slide number 9 or slide number 10?
It's going to slide number 9.
Number 9, okay. Excuse me, sorry for you all. So since we have some issue with the Microsoft team that prevent other to join us, so right now I have to advance slides for her on my system. So yeah, we're gonna be a little bit delayed. But since she going to send next slide, and that would be the way we're going to do for this talk. Thank you for your patience. Okay, and you can keep going, Elisha. Thank you.
Of course, thank you. So as I mentioned, there's an interest in growing hops in New England again. So this is a map put up by the Connecticut Brewers Guild. This is their beer trail map, and it's a map of all the current breweries in Connecticut. And with this resurgence in microbreweries in the area, there's a demand for local hops to support these breweries. So there's been increasing number of hop yards. In 2018, the Connecticut Hop Growers Association estimated that there are at least 45 acres of hops growing in the state at that time. Next slide, please. So in order to help serve this growing industry, I'm just waiting for the delay, sorry. Yep, okay, so in order to serve this growing interest in growing hops, Jim Lamondia established research plots to study how hop production would work in Connecticut, which cultivars would grow well here, what are the different management practices needed, what are the potential disease and insect pressures. And we published the first 5 years of this study in a hop bulletin in May of this past year. That goes through all the details of that. Next slide, please. So for that study, we established 2 hop yards. The first one was established at the Valley Laboratory, which is in Windsor, Connecticut, and that was set up in 2013. Next slide, please. And the second was at the Lockwood Farm, which is in Hamden, Connecticut. Next slide, please. There initially had 5 varieties that were used in the study. They were Alpha Roma, Cascade, Newport, Pearl, and Summit. And these were chosen for different flavor profiles, but also because of their different resistances and susceptibilities to 2 of the most prominent fungal pathogens of hop, which is downy mildew and powdery mildew. Since the hop yards were established, there's an additional 16 varieties now at the Valley Laboratory and an additional 47 varieties at Lockwood Farm. Next slide, please.
So,
all right, so the hop yards were managed similarly to that of commercial growers. So in the early spring, we would tie up the hop vines and we would train 1 to 2 main vines. Next slide, please. The hops were managed similar to commercial growers once again. So once the hops reached about 6 feet in height, then the lower 3 feet of the vines would be stripped of all leaves, and that decreases disease pressure. Throughout the growing season, the hops would be monitored for pests and diseases, and they would be sprayed with fungicides or insecticides as necessary to control whatever was occurring. Fertilizers were applied throughout the growing season, and there was continual weed management as necessary. Next slide, please. So at the end of the growing season, which is depending on the hop cultivar, anywhere from mid to late August to early September, the hops would be harvested. And you can see here an image of our hop harvester. And this will, as we cut off the vines, it will strip the cones from the vines so we can collect all the cones. At the end of the growing season, these cones would then be processed, they'd be dried down, and we would assess them for yield and for quality. And that is part of that 5-year study I mentioned earlier. Next slide, please. So throughout the growing season, we were assessing diseases, as I mentioned. The most common disease that we see in the Northeast on hops is downy mildew. So this is a pathogen that infects the leaves, the buds, the shoots. It leads to significant yield loss within a growing season if left uncontrolled. It really loves the humid weather that we have in the Northeast. And unfortunately, once you get it on your plant, it can actually get down into the crown and buds of the hop rhizome. And so it will become a perennial disease that you'll see every single spring. The other disease that we have, next slide, please, that's prominent is powdery mildew. And we actually saw it for the first time in Connecticut in 2018. We found it on a hop yard in southeastern Connecticut, and we have not yet found it on the research plots that we work on. So it seems that although it can be found here in Connecticut, it's not a widespread disease at this time. Next slide, please. But what we did find was in 2018, we observed a new disease that we had not yet seen before. So this was first observed in July at the Valley Laboratory. We found it on 8 cultivars: fungal, willamette, halitaur, chinook, galena, alpha aroma, pearl, Newport, Saz, and summit. We found it mostly on the leaves. There was also a few cones infected. And then later on, we found it in early August on all of the cultivars growing at Lockwood Farm. Next slide, please. So symptoms of this disease on the leaves, they'll often start as like a water-soaked lesion, but then they will turn into an ellipsoid brown-gray lesion. I think you can kind of see white concentric rings. So in this figure, the image on the right-hand side shows more of those rings, very pronounced. It has a chlorotic margin around it. Later in the summer, you can see black panidia being produced on the adaxial side of the leaves. And under hot and humid conditions, it will start to ooze a conidial mass. These lesions, the brown-gray lesions, they're not delimited by veins. We often see them associated with leaf margins, but not always. Next slide, please. As I mentioned, we also saw infections on the cones. So this would be like a dark reddish margin around the bracts. And this is important because once the cones have this damage on them, they can no longer be used for growing. So they will lead to yield loss of your crop. Next slide, please. So we isolated the sample and we grew it on half-strength potato dextrose agar. The morphology of it on the plates is, as you can see on the left-hand side, are fluffy white mycelia. They kind of have like a gray banded ring to them. They're irregular in form with undulate margins, and they have a flat elevation. On the right-hand side, you can see that pycnidia are also produced in culture. They tend to be a little larger in culture than they are on the leaf surface. They look black, they're sparse, and they're regularly dispersed on the surface of the agar plate. Next slide, please. So this is a cross-section through a pycnidium. You can see that the pycnidium is conical to globose or flask-like shape. It's brown to dark brown in color and does have an ostiole. These are found subepidermal on the leaf and petiole tissues of a hop plant, and they do erupt through the surface, and they're also unilocular. If you look around the edge of the pycnidia, you will see that the cnidiophores are on all the inside surfaces of the pycnidia. Next slide, please. So the cnidiophores are non-septate. They are hyaline and unbranched, and they're determinate. And those are on the left-hand side of this figure. On the right-hand side, you can see the alpha cnidia. They are hyaline. They're mostly E-gutulate, although sometimes they can rarely have one or more gutulations. They are single-celled, although often you'll see a constriction in the middle, which makes them appear dumbbell shaped. And they're cylindrical or clavate, and you can see they have obtuse ends. We did not observe a sexual state for this fungus, nor did we observe any beta or gamma cnidia, only alpha cnidia. Next slide, please. So based on the symptoms we saw in the field, as well as the pycnidia and the cnidia, this very closely matches another hop pathogen called Phoma exigua, which causes phoma wilt and has been reported on hops in China, Europe, and New Zealand. An older synonym for Phoma exigua is Phyllostichta decidua, and that was reported as causing hop disease in Wisconsin in 1944 and in Iowa in 1929. Next slide, please. Recently, Phoma exigua was also reported as causing disease in Slovenia on hops. There have also been other closely related Phoma species reported as causing hop diseases, such as Phoma ileana in the Netherlands, Phoma macrostoma in Poland, and Phoma herbarium in Spain and China. So initially, we thought this might be a Phoma species. However, our pycnidia wasn't quite the same morphology as the Phoma species described, and our other cnidia were a different size than the ones described. So we weren't sure if this was going to be a new species of Phoma infecting crops. Next slide, please. So we took 3 isolates. We have Connecticut 2018-1, which comes from an alpha aroma leaf from the Valley Laboratory hop yard. Connecticut 2018-2, which is from the variety Saz, and it's the cone tissue that we sampled. And Connecticut 2018-3, which is from a leaf of the cultivar Newport that was taken at Lockwood Farm. And all of these are plated onto half PDA. We did hyphal tip isolation, and we also took single cnidia to start some pure cultures. We use these to perform Koch's postulates on strap cuttings of the hop inside of the greenhouse, inside of growth chambers. And we were able to confirm that these fungal strains do infect hops and cause similar symptoms. And when re-isolated, they match the morphology of what we had inoculated with. And our uninoculated controls showed no disease symptoms. We then took these purified cultures and we sequenced them. We sequenced the internal transcriber spacer unit of ribosomal DNA, the histone 3 gene, the calmodulin gene, the elongation factor alpha 1A gene, beta tubulin, the 28S or the large ribosomal subunit, and also RNA polymerase 2 subunit. When we did blast matches for these sequences, they did not have any close matchage with Phoma. They actually match more closely with Diaporthe species. Next slide, please. So we then ran a maximum likelihood phylogenetic analysis. This included 52 sequences representing 41 taxa. And we included Diaporthe species and also Phoma species, including those that have been previously reported on hops. And so within the large red rectangle are Connecticut isolates. They grouped very strongly together. And they also clustered within the Diaporthe group and far away from the Phoma species group. So based on this evidence and the differences in morphology, we believe this is a member of the genus Diaporthe, and it's not a Phoma, so it's not one of the previously reported hop pathogens. Next slide, please. So in order to better classify where this new fungi is within the Diaporthe, we then did Bayesian interference using 5 of the genes, so ITS, calmodulin, elongation factor alpha-1, beta-tubulin, and histone. We only used those 5 genes because they had the most representative sequences for those genes, whereas 28S and RPB2 didn't have as many representative taxa, so it wouldn't have given us as much information using them in a tree. So for this tree, we had 95 taxa represented and 159 sequences. And next slide, please. So here you can see in the red box, the Connecticut isolates all clustered very strongly together with 100% bootstrap support. It looks like 2018-1 and 2003 might be somewhat slightly more similar than they are to 2018-2, but they're still strongly a group unto themselves. Next slide, please. Although Connecticut isolates do differentiate from other Diaporthe species, they don't have any other taxa that they were highly similar to. So next slide, please. This is just zooming in on the node which breaks off our Connecticut isolates from the rest of the Diaporthe that they're closely associated with too. And you can see that there's 94% bootstrap support for them being a separate group from the rest of the Diaporthe within this region. So that's showing us that they most likely are a new, previously undescribed species. Next slide, please. So from this outgroup, actually now I think I'm caught up with you guys are. So when you look at this box, this is just showing that after that breakdown is 94%, the next closest group is a 74% match. This is a very large group. It includes Diaporthe ambigua, Diaporthe endolete, Ordinaria, Arctica, Capite, Lucanica, Novum, Infecunda, Batata, Citrus, Melonis, Sojae, Convolvuli, Endophytica, Physiolorum, Shiny, Ticornaire, Tenoranthifolii, Helianthi, Hordiae, Vexins, Megalosporia, Gangia, Manihotia, Longospora, Sclerotoides, Matini, Rhinochaetoporum, Oxyperinesis, and Brasiliensis, as well as 5 different Diaporthe species that Gomez et al. In their paper described as different species from the other Diaporthe, but not yet classified. So although they are somewhat similar to these species, they are definitely a distinct group. All right, next slide, please. So in order to differentiate our new species, so we have our genetic analysis, which shows that it's its own group. It can also be differentiated morphologically because it only produces alpha canidia. It has unbranched canidiophores. The alpha canidia mostly are egutinate, rarely with one to many gutylations. They're cylindrical or clavate with obtuse ends, and some are constricted in the middle. And also by the size of the canidia. Most of the other closely related species that could be confused with it have larger canidia. And in order to further differentiate it from the Phoma species that we know infect hops, those ones, in addition to being genetically different, the Fulma species have a sclerotinized parenchyma around the peak media, and our Diaporthe species lacks that. And also the alpha canidia in this Diaporthe species are smaller than those of the Fulma species previously described. Next slide, please. I know there's a bit of a delay, so I just want to double check that you guys are on the slide with the plant disease cover now.
Yeah, I think we are.
Okay, perfect. I just wanted to make sure. So based on all this evidence, our genetic evidence and a morphological evidence, we propose this as a new Diaporthe species. We have named it Diaporthe humulicola. Humulicola coming from the host, Humulus lupulus, and cola meaning one that grows on. We propose the disease to be called Diaporthe leaf spot, and that's based on the symptoms on the leaves of this our hop plant. And we recently published it this year in Plant Disease. And I just wanted to point out Michelle Salvis, who's our research technician, her beautiful picture of the disease hop cones made it to the cover of Plant Disease, which we were really excited about. So this, all right, next slide, please. So we initially observed this, like I said, in 2018, and we've continued to find this pathogen each subsequent year on both hop farms. In 2018, it appeared in July at Valley Laboratory, in August in Lockwood Farm. In 2019, it appeared as early as June. And then this past year, it was observed in July again. Next slide. So looking at what might be the environmental conditions increasing the likelihood of infection of this pathogen, this graph here shows daily temperatures for the Valley Laboratory yard. On the y-axis is average temperature in Celsius, and along the x-axis is the day and month. The orange line represents 2019, and the blue line represents 2018. The blue arrow shows when infection began in 2018, and the orange arrow when infection began in 2019. We don't have this data yet for 2020, which is why it's not included on the graph. So as you can see, infection started earlier in 2019. There was maybe slightly warmer temperatures, but they were very similar in temperature for both years. But we don't see disease until we do get that warmer weather. So we know that the disease is favored by higher temperatures. Next slide, please. But we did observe, this is looking at cumulative rainfall at Valley Laboratory. And once again, on the y-axis, we have rainfall in centimeters. And on the x-axis, we have month and day. The blue line, once again, is 2018, and the orange line is 2019. So here you can see that 2019 was a much wetter year than 2018, and that when they're hit of almost 60 centimeters of rainfall is about when we saw the disease at both locations. So this really suggests that this pathogen, although it does need warm temperatures, also really needs leaf wetness in order to increase in severity. So this is something that we're going to continue researching and looking into, especially for things like timing of fungicide application or different cultural practices that might help reduce this disease and manage it. Next slide, please. So a continuing part, now that we found a new disease, is figuring out how are we going to manage this disease. As you've seen in the previous images, the disease does infect cones. And once it infects the cones can't be used in the brewing industry. Also, potentially, if the foliar disease gets bad enough, there might be reduced hop production in general. So this might be a double way that it could be affecting hop yields. So We believe that based on the biology we can see so far, that a lot of management practices, cultural practices aimed at reducing leaf moisture will most likely help decrease the spread of this disease. Also, fertilization and proper irrigation would also help, because when you have healthier plants, you tend to have less disease. And lastly, application of fungicides registered for hop in Connecticut could help control this disease. This information we did include in the guidelines for integrated pest management for hops in Connecticut, which we put out in both 2019 and 2020 with this disease, to help our local hop growers better identify and manage it if they do see it in their hop yards. All right, next slide, please. So to figure out which fungicides might work well, we did a trial of fungicide efficacy in vitro. So we amended half-strength potato dextrose agar with our different fungicides. We calculated rates that were based off of the recommended field rates for the different chemistries. And this was our way of initially screening which might help control this disease. So in the figure on the y-axis, we have relative mycelial growth. So that's looking at how well our Diaporthe isolate grew on an unamended plate, so no fungicide, versus a plate amended with a fungicide. And this is the ratio of how much was able to grow on the fungicide plate. We tested this for two different isolates, Connecticut 2018-1 and Connecticut 2018-3. So one from the Valley Farm and one from the Lockwood Farm. And along the x-axis, you can see the concentrations we chose. So for our first fungicide, which is trifloxystrobin, this is the commercial name is Flint. It's registered to control hop powdery mildew. You can see that even at the lowest concentration of 10 ppm, it reduced fungal growth by almost 50% for the Connecticut 1 isolate, and all the way down to 20% for the Connecticut 2018-3 isolate. So we did see control with this chemistry. Next slide, please. The second chemistry we tested was pyraclostrobin and boscalid mixed, and this was the chemical name of Paget intrinsic. And you can see that there is a very high inhibition of fungal growth. So at only 1 parts per million, it reduced growth of the 2018-1 isolate down to 2.5%, and it completely inhibited growth of the 2018-3 isolate. Next slide. So we tested Paget, but the equivalent that's registered on hops is pristine. Pristine is registered to control both powdery and downy mildew. And it's also registered to control Phomopsis and other crops, Phomopsis being the alternate name to Diaporthe. So pristine might be a very good option for Connecticut hop growers. Next slide. So we also tested pyraclostrobin alone to see if it was effective without the boscalid. This is trademark name Insignia, and you can see that it did inhibit the fungi. At 0.6 concentration, it reduced it by to 5% for Connecticut 2018-1, and down to 2% for Connecticut 2018-3. So it does have control, not as high of control as with the boscalid, but still high control. Next slide, please. So one thing, the insignia is actually not registered for hop in Connecticut. So this was purely just for us to test whether or not boscalid was necessary in the chemistry. Next slide, please. So for our potential management strategies then, for those fungicides, right now we would probably want to pursue looking at pristine, so pyraclostrobin and boscalid, as a fungicide to help reduce Diaporthe, especially since it could be sprayed on hops right now for downy mildew and powdery mildew. It might make sense to include it in the rotation to help control this disease as well. But this will be evaluated in the field before we can make any stronger recommendations for its use. All right, next slide, please. So with that, with our new disease, we know that we have a new pathogen in Connecticut, and it can greatly reduce hop yields. Potentially, it can damage cones as well as the leaves. And it's recurring each year. since its establishment in our hop yard. So we will continue to be studying this. The picture on the left, for those of you who have not yet met him, is Shrikant Kodati, who is the new postdoc with Jim Lamandia at the Valley Laboratory, and he is working on the HOP project. And together we are continuing to study more about the biology of our Diaporthe Pathogen, its disease cycle, and looking at potential management strategies. And we are greatly looking forward to when we can go back to having field days. This is an image of one of our field days with the Connecticut Hop Growers Association. And we can talk with growers about this new disease and what we're finding and give them some strategies to help manage it in their field. So next slide, please. So with that, I would just like to thank all of the authors who contribute to this project, as well as Michelle Salvis, who you can see on the far left image, who's our research technician, Katja Mara, who was a previous postdoc before I arrived and started a lot of the hop research, as well as Nathaniel Child, who also was a research technician with Katja Mara and helped start all this research work. In the center is Jim Presti and his team who helped maintain the hops at the Valley Laboratory. And on the right-hand side is Rich Ciccarelli and his team at Lockwood Farm, who manage the hops there. And for our funding agencies, we have the USDA, the Connecticut Department of Agriculture, the Association for Developing Hop Varieties, and the Connecticut Agricultural Experimentation. And next slide, please. With that, I'd just like to thank all of you for tuning into this presentation. I hope you enjoyed it and learning about our new pathogen. And are there any questions?
Thank you so much, Elisha. That's very interesting presentation you have on the growing hop in Connecticut. Right now, if you have time, feel free to type any question in the Q&A session on the upper right corner of your screen. Now we have two questions, one from Rob. He asking, what is the rootstock resistance?
So I'm assuming this is referring back to that slide I showed of the 5 different cultivars that we're growing, and their different resistance to downy mildew, which Cascade has rootstock resistance. So what that means is that there's actually a resistance of the rootstock to the parination of the downy mildew within the rhizome, but the upper leaf plants will get infected during the year. So for instance, if the Cascade is planted next to Alpha Roma and Alpha Roma has the disease down into the rootstock, then in the spring it's going to put up shoots, which we call basal spikes, that are infected, and then that infected material can then get onto the Cascade, and the Cascade leaves will get infected with it. But it has resistance where it's less likely for that disease to get into the Cascade rootstock and continue the disease year after year.
Elisha, and the next question is someone asking, what are the most important traits in hop breeding, horticultural or pathological?
That is a really good question. I'm not a hop breeder, so they might have a different view than I do. I think a lot of the work that goes into hop breeding is looking at different flavor profiles. So as beer trends change, so for a while it was IPAs, and then it was England IPAs, things like that, that they're looking for different flavor profiles within the hops, and so that can drive a lot of the breeding. But there is also work being done for disease management. So, in the Pacific Northwest, which is the main hop growing area, they are spraying more for powdery mildew and downy mildew. So if you have more resistant cultivars, you can do less sprays. That's always a good thing. So that's probably kind of the main things with breeding. There might also be some interest in breeding better for growing the hops in different regions. So there's hops that grow really well in England that are very much adapted to their climate, that they actually don't share with outside regions, some of those varieties. So it could be interesting to do breeding to try and increase hop yield and productivity in different climates as well.
Thank you so much, Elisha. Look like we have a lot of questions. The next question is, Why hops are mainly grow in PNW? East Coast are too humid?
So the main reason why is because when downy mildew came in to the Northeast and almost wiped out hop production.
Excuse me, look like I'm just lost her. Sorry, you all. Let me call her back. Sorry about that. This is some technical issue with the system. Hold on. Let me call her back. Hello, it's okay,
Sorry
Alicia.
about that.
Don't worry.
I don't know what happened.
Don't worry. This thing happen, people when some people work from home and lockdown, sometimes the network overused. So let me re-question again. Why hops are mainly grow in PNW? East Coast are too humid?
Yeah, so basically in the Northeast they were grown for a long time, but when downy mildew came in, it basically wiped out the industry here, and that's why it moved to the Pacific Northwest because they are drier, and so it's downy mildew wasn't as rampant there, and so they were better able to grow them in that region for that reason. But there's really no reason not to grow hops here now. It's just getting varieties that are well adapted to our warmer, more humid climate.
Okay, thank you. And the next question is there any organic management option for this disease?
So because it's a new disease, we don't really know any management strategies for sure yet, and that's what our field work is going to be assessing. There are some organic options for controlling powdery mildew in hops, and because powdery mildew has a somewhat similar conditions that it grows in to our Diaporthe, potentially we could try testing some of those options out to see how they work on our Diaporthe species. So I would say that at this time we don't have recommendations yet other than cultural practices, but we'll be testing those organic solutions so we can have some options for growers.
Thank you. And the next question from JP. Could you speak to crop yield losses due to this pathogen?
I can. So we did not assess yield loss from this pathogen directly. We did see some decrease in hop production in 2019. And then this past year, with the severe drought that was happening in Connecticut, hop loss was really low again. And it's hard to decide if it's because of this disease or because of the drought. So we do believe that each year that we've had in the hop yard, we've seen more disease each subsequent year. And so if that trend continues, then the longer you have this in your hop yield, the more decline in production you might have. So it potentially could cause major yield losses. We haven't seen it yet, though. So this might be more of a minor issue. We really need to see it longer and at more fields before we have a really good answer at how damaging this could be to the hops.
Thank you, Alicia. And next question is, do you have any information on resistance to fungicide? How quick tanks mixtures?
That's a great question, and that is definitely something that we need to investigate further. Based on the in vitro assay I did, because we saw such a drastic decline in growth on the media plate with fungicides amended, that suggests there is no resistance at this time with those particular isolates in the lab. But that would be something that we would need to monitor because especially if you're using the same chemistries for multiple diseases, if you're getting resistance to them, then that could be an issue. So what I mean by that is if you're spraying a chemical early in the season for downy mildew, and then you're spraying it later in the season for Diaporthe, you're exposing potentially that fungi to that chemistry for a longer period of time and for more applications. And so you could be pushing resistance quicker than if you're only applying it once or twice a year for one disease. So it's definitely something that needs to get monitored and checked. The question about tank mixes, that's another potential reason to use the pristine because it is a tank mix of pyraclostrobin and bosclid. So there is less of a chance of resistance developing potentially, but that really needs to be evaluated in the field and monitored.
Okay, and the last question is, did you grow tea makers hop variety? I'm curious if it's resistant to powdery and downy mildew.
We did not grow that variety. If you look online, especially if you go to wherever the rootstocks are being sold, they should have information about resistance and susceptibilities to those two diseases. We have found slight differences in variety susceptibility here versus other growing regions where a lot of the rootstocks are coming from. So for instance, Cascadus was pretty resistant to powdery mildew, but we did see it in Connecticut on Cascade at a much less amount than on other cultivars, but it still suggests that here it might be a little bit more susceptible. So I would start with going to wherever you're buying the rootstock from and seeing what they suggest for susceptibilities and resistance, and that should be a pretty good guideline. And maybe it will be slightly different here, but it should be still pretty. Even if there might be a slightly more susceptible here, it shouldn't be drastically different than what they're recommending.
Thank you so much, Elisha. Looks like we don't have any further questions. And once again, thank you so much, Elisha, for giving us the very nice presentation on your research at our farm last year or years before. So it's very useful. And again, we are sorry for a little bit technical issue that we have due to the Microsoft Live. I already sent the technical question to the person who take care of the system, and I wish we can fix the issue for the longer term. But thank you again, Elisha, and I will give her a big applause for her talk, and everyone can give her the virtual applause at the end. So thank you so much, and I will see you for the next talk in the next couple of weeks. Thank you so much.
Thank you so much, everyone.